The architecture of the human brain spans multiple scales of resolution, from microscopic genetic instructions to macroscopic systemic behaviors. The challenge for neuroscientists is to connect these domains, to understand how biological mechanisms at the smallest level assemble the circuits that generate thought and action. That structural assembly begins before the brain itself even exists. It starts in the genome. Every clinical observation of a patient's behavior ultimately traces back to the specific execution of these genetic blueprints. This diagram outlines the human genome, which consists of about 20,000 genes. Of those, roughly 14,000 are expressed in the nervous system, and a distinct subset of around 6,000 are entirely exclusive to it. This is a lateral silhouette of the human brain, mapping mRNA expression. The locations and varying levels of expression for specific genes dictate the physical construction of the organ from region to region. Because the macroscopic structure relies heavily on macroscopic genetic precision, a single point of failure at the cellular level cascades into severe anatomical anomalies. A mutation in the ASPM gene, which regulates mitotic spindle function during cell division, halts typical development. The resulting condition is microcephaly, characterized by a severely reduced brain size and distorted structural organization. An error at the scale of a single mitotic protein disrupts the scaling of the entire organ. To understand the brain's form, analysis must begin at the genetic level. Moving up from the genetic code, those instructions build the primary computing units of the nervous system, nerve cells. But for decades, the physical boundary of these cells was a mystery. Early microscopes lacked the resolution to separate the dense tangle of nervous tissue. Biologists like Camillo Golgi promoted the reticular theory, concluding the brain was a single continuous protoplasmic net. Santiago Ramon y Cajal proved that assumption wrong. Using Golgi's own staining techniques, Cajal championed the neuron doctrine, demonstrating that nerve cells are discrete, independent units communicating across physical gaps. This physical independence means individual neurons can adopt extremely diverse shapes. In the nervous system, a cell's morphology strictly limits its exact computational role. This drawing traces a single cerebellar Purkinje cell. The massive tree-like dendrites create immense surface area for high convergence, receiving inputs from thousands of neurons. By contrast, the single thin axon extending from the base relays the integrated electrical signal out to distant targets. A neuron's physical geometry operates as its hardware. The branching structure directly determines how it processes information within a local circuit. While the neuron doctrine established that these cells were independent, confirming the physical gap between them required magnification that light microscopes could not achieve. In the 1950s, the electron microscope provided the resolution needed to finally observe the ultrastructure of nervous tissue. This electron micrograph reveals the central command center of the neuron. The large purple structures are the cell bodies, or somas, occupied by distinctly round nuclei. Moving outward into the tissue, we can trace the thick apical dendrites threading their way through the complex extracellular environment to receive incoming signals. Following the transmission pathway, this cross-section shows the central axon in blue. It is wrapped tightly by a laminated myelin sheath, shown in gold, which acts as insulation to speed up electrical propagation. At the very end of the axon, we find the chemical synapse. When an electrical signal arrives, spherical synaptic vesicles fuse with the membrane, releasing neurotransmitter molecules directly into the physical gap between cells. The synaptic cleft is the critical bottleneck of the nervous system. It is the exact location where an integrated electrical signal is converted into a chemical message. Maintaining a massive dendritic tree and continuously trafficking vesicles down the length of an axon demands a highly structured internal transport system. This grid maps the cellular cytoskeleton. Tubulin, shown in green, forms the stable main branches of the cell body and dendrites. Actin, highlighted in red, drives dynamic growth at the very tips of the axonal and dendritic processes. Neurons do not sustain this extreme metabolic activity in isolation. They rely on glial cells, such as astrocytes, to regulate the surrounding ionic and chemical environment. A functional neural circuit requires both the precise internal scaffolding of the individual neuron and the external biochemical regulation provided by the surrounding glia. Isolating single cells is useful for structural understanding, but clinical neuroscience requires observing millions of these circuits operating simultaneously inside a living subject. Early structural imaging relied on X-rays. While these captured dense bone, they failed to resolve the soft tissue nuances of the brain. Early functional measurements yielded better results. Electroencephalography, or EEG, placed electrodes on the scalp to capture the aggregate electrical synchrony of the cortex. EEG proved that neural activity could be recorded externally, but it lacked the spatial resolution required to pinpoint exactly which circuits were firing. In the 1970s, computerized tomography solved the structural problem. By taking serial X-rays from multiple angles, CT scanners allowed clinicians to differentiate soft tissue layers. CT scans provide excellent three-dimensional mapping for detecting tumors or gross pathologies, but they capture static anatomy. They cannot reveal brain function. Where fMRI tracks vascular changes, positron emission tomography injects a radioactive tracer to visualize specific metabolic processes, tracking direct receptor binding at the synaptic cleft. When functional imaging shows a region of the brain lighting up, the clinician directly observes the aggregate metabolic demands of the microscopic synapses we looked at earlier. Clinical neuroscience relies on linking structural anatomy to systemic data processing. This is a whole brain tractography profile. It maps the dense white matter communication highways connecting disparate regions of the cortex. These massive tracts are the final macroscopic product of the precise cellular blueprints and regional genetic mapping established during embryogenesis. Every thought, action, or systemic pathology captured by clinical imaging originates entirely from this cellular baseline. Understanding the nervous system requires seamless integration across scales, moving continuously from the expression of a specific gene to the specific morphology of a neuron to the systemic behaviors observed in a scanner.